Post-transcriptional modifications. Expression of membrane and secreted immunoglobulin M. An immunoglobulin can exist in two forms, secreted or membrane form. These forms differ in the amino acid sequence at the carboxyl terminal region of the heavy chain. In the secreted form, the carboxyl terminal domain contains a hydrophilic stretch of about 20 amino acids. The membrane form has a 40 amino acid sequence with a hydrophilic region outside the cell, a hydrophobic transmembrane segment, and a short hydrophilic segment in the cytoplasm. A single B cell can produce secreted or membrane-bound forms of a particular immunoglobulin. Inside the nucleus of a B cell, the rearranged heavy chain gene contains 2 polyadenylation sites in the C-mu segment and 2 in the C-delta segment. PA-mu-S is located at the 3' end of the C-mu-4 exon, while PA-mu-M2 is located at the 3' end of the mu-M2 exon. If polyadenylation occurs at PA-mu-S, the remaining introns are spliced out, producing mRNA encoding the secreted form of the heavy chain. If polyadenylation occurs at PA-mu-M2, RNA splicing removes the mu-S sequence at the 3' end of the C-mu-4 exon along with introns, producing mRNA for the membrane form of the heavy chain. The mRNA encoding the secreted and membrane form is exported out of the nucleus to be translated by ribosomes into secretory or membrane-bound antibodies. These antibodies have paratopes that specifically interacts with the same antigenic epitope. Simultaneous expression of immunoglobulin M and immunoglobulin D. The rearranged heavy chain gene contains 2 polyadenylation sites in the C-mu segment and 2 in the C-delta segment. PA-mu-M2 is located at the 3' end of C-mu-M2 exon and PA-delta-M2 is located at the 3' end of the C-delta-M2 exon. IgM is produced if polyadenylation occurs at PA-mu-M2. The mu-S exon along with introns are spliced out, producing the mRNA encoding the membrane form IgM. IgD is produced when polyadenylation occurs at PA-delta-M2. RNA splicing removes the C-mu exons and delta-S exon along with introns, producing the mRNA encoding the membrane form IgD. IgM and IgD produced then attaches to the membrane of the B cell.